Hey guys, and welcome to AP Help 101. So, this week I'm going to talk about meiosis, and last week I talked about mitosis, and meiosis is a little bit different from mitosis. It's the division of sex cells, eggs and sperm. So, just a brief introduction onto what meiosis is. Basically, it's the process that produces ova and sperm, which are our gametes, or sex cells. And gametes are haploid cells, which means they have half the number of chromosomes of a parent cell. So instead of having 23 pairs of chromosomes, they really only have 23 chromosomes. There are two parts to meiosis. We have meiosis 1 and meiosis 2. Meiosis 1 is the part that's different from mitosis. It's reduction division, where our homologous chromosomes separate, and this is when crossing over occurs. And I'll go more into details as to what happens in meiosis 1. And meiosis 2 is very similar to mitosis. It's just the separation of two cells. It's the sister chromatids separate into different cells. So meiosis 1. Now, I'm going to list these in steps, or each of the stages, but it's not necessary to memorize what happens in each stage. The College Board changed that, so you don't need to memorize what's in each specific phase, but it's important to know what happens in general in meiosis 1. So the first stage, prophase 1, this is the pairing of the homologues, and crossing over takes place, or the exchange of small parts of chromosomes. So what that means is you have your dad's chromosome and your mom's chromosome, and certain genes will switch. They will switch places on both of the chromosomes, and then you'll have a mixed version of your parents' genes, which is why you look a little bit like your dad and a little bit like your mom. Then we have metaphase 1, where the chromosomes line up and the spindle fibers connect from the centromeres to the poles of the cell. Anaphase 1 is then the separation of the chromosomes, and they move to opposite poles. And then telophase is the continuation of separation. And then, of course, cytokinesis is the split of the cytoplasm, and it technically occurs during telophase. Sometimes in some organisms, there is an interphase between meiosis 1 and meiosis 2. It doesn't happen in all organisms. Humans don't have this interphase between the two. We just go from meiosis 1 to meiosis 2. We don't have that interphase in between. Meiosis 2. This is very similar to mitosis. The same process happens. The remaining chromosomes line up and they are separated to opposite ends of the cell. And as a result of meiosis completely, of 1 and 2 together, we have 4 daughter cells with only half the number of chromosomes from each parent cell. And all 4 of these are different due to the crossover that takes place. That's why you don't have exactly the same genetic code as either one of your parents. You have a mix of what they have. So this is the difference in meiosis and mitosis, because in mitosis you have 2 daughter cells only, and they're identical to the parent cell, whereas in meiosis you have 4 daughter cells, and they are completely different from the parent cell. So here's just a diagram of what happens in meiosis. We have the first meiosis 1 at the very top, so you can see we have our parent cell. Prophase is when we have our crossing over, and you can't really see that so much on the diagram, I guess if you can zoom in. But there's crossing over in prophase 1. In metaphase 1, you'll see that all the chromosomes are lining up. And then in anaphase 1, there you can see the crossing over that took place right there. You'll see the red and the blues have switched. And then in anaphase 1, you'll see them moving to opposite poles. Telophase and cytokinesis, you have the 2 different daughter cells. And then from there, you continue to meiosis 2, so they'll show you metaphase 2, and that's just lining up of chromosomes yet again. And then anaphase 2, where they separate out. And finally, telophase 2, where you have 4 completely different daughter cells, which only have half the genetic makeup of the parent cells. So a lot of genetic variation comes about from meiosis. There are actually 3 types of genetic variation that comes out from meiosis. The first one is independent assortment of chromosomes. So in meiosis, there's a 50% chance that the gamete will receive the maternal chromosome and a 50% chance that the gamete will receive the paternal chromosome. And so that's what varies everyone, because not everyone will have the maternal, not everyone will have the paternal. It's going to be a little bit of both. Crossover is also another form. It's one of the main forms of genetic variation, actually, and that's because it creates recombinant chromosomes. So we combine genes from both parents, and oftentimes about 2 to 3 crossovers takes place. So it's not just one gene that's being switched; it's multiple genes that are being switched, which gives you the makeup of both your mom and your dad, which then leads to more variation in the population. And finally, there's random fertilization. So, really, they actually did the math. There's 8 million possible chromosome combinations. So, just imagine the number of recombination possibilities there could possibly be. And that's why each one of us looks so different from each other. If you have siblings, yes, you do share much of the same genetic makeup if you come from the same parents, but sometimes you look nothing alike each other, and it's all due to independent assortment of chromosomes, crossover, and random fertilization, because there's all these different combinations that could come about in because of meiosis, actually. And finally, the last thing I'm going to talk about is the timing of the cell cycle. So we actually have a cell cycle control system in our body, which regulates the rate at which cells divide. And there are 3 checkpoints during the cell cycle. The most important checkpoint, I think, is the G1 checkpoint. And basically, if a cell passes that G1 checkpoint at the G1 stage, the cell will most likely complete division. If it does not pass that checkpoint, it will go to a G0 phase, and that's basically it just stops growing. There's no point in that cell anymore. And the G2 and the M stage are just growth checkpoints to make sure that everything is going smoothly, that everything's being created correctly. And again, if it passes that G1 checkpoint, it's most likely going to continue and complete cell division. So that is meiosis, guys. Really, a quick rundown of it. If you have any questions, comments, suggestions, leave them down in the comments below. And I will see you guys next week.